Hey guys, Miss Cote here, and welcome to our next unit, which is cell communication and cell cycle. To start off this unit, we're going to look at the different ways cells can communicate with each other and look at how organisms can use cell signaling to maintain homeostasis. This first episode will go through the basics of cell signaling, focusing on different methods of reception and transduction. So let's get started. One of the most classic examples of cell communication and cell signaling is the fight or flight response. The body's response, whether you fight or flee, is to break down glycogen into glucose and convert that into energy. And that response will happen because of this guy here, which is called epinephrine, or otherwise known as adrenaline. This is the signal that will start the relay to get your body more glucose. Even the simplest cells and organisms have the ability to communicate with each other. And a lot of what we know about signal transduction pathways has been through studying these cells. For example, studies in yeast show that unicellular eukaryotes can identify mating partners by secreting signals. The cells of different mating factors can find each other by secreting their specific factors, A or alpha. And when the factors are recognized by receptors, the two cells will mate and produce a new yeast cell that has both mating factors. Even bacteria can communicate with each other in a method known as quorum sensing. By monitoring their density, how many bacteria there are in a particular area, these bacterial cells can determine if they have the numbers to launch an attack and make you sick. They're not going to bother if they don't think they'll be successful. There's a really great TED Talk by Dr. Bonnie Bassler about quorum sensing. I'll post it to our website if you would like to watch it. In multicellular organisms, cells use signals to talk to each other. Sometimes these signals don't need to go very far, and other times they may need to travel great distances to deliver their message. In some local signaling methods, cells that are directly touching each other can talk by passing signals back and forth through their cytoplasm. Remember gap junctions and plasmodesmata? One cell can produce a signal which can pass through these channels and deliver a message to the adjoining cell. Another type of direct contact between adjacent cells would be cell-cell recognition, where a surface protein on one cell, representing the signal, would match and bind to a surface receptor on the next cell, causing some message to be delivered to the cell. Local signaling also includes signals that only need to travel short distances, also called paracrine signaling. Here we have a secreting cell. Let's say it's secreting growth factors, and those signals are being picked up by neighboring cells, also called target cells. Another example of local signaling that's used in the nervous system is synaptic signaling. When a signal is being passed from neuron to neuron, there's a small space or gap between neurons called a synapse. In order to pass the signal, the first neuron will secrete chemical signals called neurotransmitters, which will diffuse across the gap and bind to receptors on the next neuron, continuing the signal. In long-distance signaling, the chemical signals are called hormones. In animals, this is the endocrine system. Plants also need to communicate long distances too. Think from roots to leaves. And although plants don't have an endocrine system per se, they're still referred to as hormones, although some people in textbooks are slowly getting away from using that term for plants. In this example, we have an endocrine cell, and the hormone is released into the bloodstream. From there, it will travel through the body until it reaches its target cell, which has a specific receptor to recognize the signal. From there, it will deliver the message. A lot of what we know about cell signaling comes from research of epinephrine and how it's used in the fight or flight response. Scientists have worked out that signal transduction can be separated into 3 stages. First is reception, where the target cell detects a signaling molecule that binds to a receptor protein on the cell surface. The second step, transduction, is when the binding of the signaling molecule changes the receptor and initiates a signal transduction pathway. This usually happens in a series of steps using different molecules to ultimately bring about a specific response, which could be pretty much anything the cell needs to do. Most of the time, it involves the transcription of a gene to make a protein that the cell needs. So first, let's talk about reception. Just because there's a signal, that doesn't mean it'll work on any cell. Cells that have the ability to recognize and respond to a signal do so because they have a specific receptor molecule that can bind to the signal. Like an enzyme substrate complex, the signal molecule, referred to as a ligand or ligand, and the receptor fit together like a lock and key. There needs to be a perfect fit in order for anything to happen. There are two categories of receptors: receptors embedded in the plasma membrane and receptors that are found inside the cell. Most signal receptors are plasma membrane proteins. Plasma membrane receptors are integral proteins that span the cell membrane. The ligands they're associated with are referred to as water-soluble, meaning that these signal molecules cannot pass through the plasma membrane, so they need to meet their receptor on the cell surface. Let's go through the three main types of membrane receptors. G-protein coupled receptors are the most abundant of these plasma membrane receptors. The receptor is a transmembrane receptor that works with the help of a peripheral protein called a G protein. When the ligand binds to the receptor, the inactive G protein slides over to the receptor and binds GTP to become active. The now active G protein slides over to an inactive enzyme and activates it, starting a chain of events, a signal transduction pathway, which we'll discuss in the next section, leading to some type of response. When it's finished doing its job, the G protein becomes inactive again and waits for the next signal. Next are the tyrosine kinases. If you look at the name, it ends in an ASE, so that means it's an enzyme. Kinases are enzymes whose job it is to phosphorylate things, and these tyrosine kinases will attach phosphates to tyrosines, hence the name. In its inactive form, tyrosine kinases are found as two monomers. When two ligands come around and bind to both monomers, the monomers will slide together to form an active dimer. The next step is to phosphorylate the tyrosines, which is done through ATP, giving up its third phosphate, and the phosphates attaching to the tyrosines. This makes the kinase primed and ready to do work. These tyrosines can now activate other molecules in the pathway. Please note that these 6 tyrosines can activate the same molecule 6 times, totally amplifying the pathway, or activate multiple pathways at one time. The third type of plasma membrane receptor is the ion channel receptor. Remember that ions are charged molecules that cannot pass through the cell membrane without the help of a channel protein, and sometimes these channels are closed. A ligand-gated ion channel receptor acts as a gate, which opens when a ligand binds, allowing the ions through into the cytoplasm, initiating a signal transduction pathway to get a response. And when it's done, the ligand is released and the gate closes up again. The second category of receptors are the ones that are found inside the cell, either in the cytoplasm or in the nucleus, and these are called intracellular receptors. Since these are inside the cell, the ligands associated with them are able to pass right through the cell membrane. This means that they are small or nonpolar molecules. Here we have a steroid hormone, which, since it's a lipid, crosses right into the cell, meeting up with a receptor in the cytoplasm. Binding activates this hormone receptor complex, which can then travel into the nucleus to act as a transcription factor, which can regulate gene expression by turning a specific gene on or off, usually on, to create a protein needed by the cell. Because the signal can get into the cell, it usually takes care of the transduction part of the pathway itself. Now that we're done talking about receptors, let's look at transduction pathways. They're usually paired with the plasma membrane receptors, since intracellular receptors can take care of themselves. When the ligand cannot get into the cell to deliver the message itself, it needs assistance from other molecules to get the message where it needs to go. These are usually multi-step pathways which use an assortment of molecules to get the message where it needs to go. There are 2 basic types of pathways. One signal transduction pathway is a phosphorylation cascade. We've already seen that kinases can phosphorylate molecules and prime them to do cellular work. Multiple kinases can create a phosphorylation cascade to ultimately bring about a response. Another important enzyme in the cascade are phosphatases, which reverse what the kinases do. So while kinases phosphorylate and activate molecules, phosphatases can take the phosphate group off, called dephosphorylation, and inactivate the molecule. These 2 enzymes work in tandem with each other to act as a switch. turning cellular activity on and off. When the ligand binds with the receptor, it turns on a relay molecule, which activates the first kinase. Kinase 1 activates kinase 2, which activates kinase 3, which activates the protein needed for the response. Notice the need for ATP at each step, where the third phosphate group breaks off and attaches to the activated kinase. Also notice the protein phosphatases, which eventually takes the phosphate group off and inactivates the enzyme. Another type of signal transduction pathway is the use of second messengers. If the initial ligand is considered the first messenger, and this molecule can't get into the cell to deliver the message itself, then it needs to rely on other molecules to deliver the message for it. These molecules are called second messengers. Common second messengers are cyclic AMP and calcium. Work with the epinephrine pathway uncovered an increase in a cytosolic compound called cyclic AMP, or CAMP. We now know that CAMP is a widely used second messenger in many pathways. Starting with ATP, CAMP is produced by an enzyme called adenylyl cyclase. Looking at this diagram of the G protein coupled receptor that we saw earlier, the enzyme on the right, that didn't have a name before, is now labeled as adenylyl cyclase. When adenylyl cyclase is activated by the G protein, it converts ATP to cyclic AMP. Cyclic AMP can, in turn, be the relay molecule that activates the first protein kinase to start the phosphorylation cascade. Another common second messenger is calcium. If you remember, storage of calcium is one of the functions of the smooth ER. Examples of calcium being used as a second messenger would be in muscle contraction and also in the secretion of neurotransmitters at the synapse. Here we have a signal transduction pathway where the G protein activates an enzyme called phospholipase C. Activated phospholipase C in turn cleaves a certain type of phospholipid in the membrane called PIP2 into diacylglycerol, DAG, which stays in the membrane, and inositol triphosphate, or IP3. IP3 can diffuse through the cytoplasm and acts as a ligand to open an ion-gated channel in the ER. When opened, calcium ions are released into the cytoplasm, down their concentration gradient from high to low, and the calcium ion concentration in the cytoplasm is now increased. This in turn will activate the next set of proteins in the transduction pathway, ultimately leading to a response. And the last part of the signal transduction pathway is the response, which could be a whole host of things. Responses in the nucleus usually involves a change in gene expression, turning a gene on or off. Or it could regulate activities in the cell, regulating enzyme activity, opening or closing an ion channel, or even stimulating cell division, which we're going to look at in this unit. One last thing I want to say about signal transduction pathways is that enzyme cascades can greatly amplify the response to the signal. Looking at this epinephrine example, you'll notice that at each step in the pathway, the number of activated products is much greater than the preceding step. So please keep that in mind about the importance of these multi-step pathways. And that covers the basics of cell communication. We'll be working with these concepts in class, applying them to different examples. So until I see you next, be a good human.